All right, guys, welcome to Laboratory 12. We have one more after this. Make sure you are turning in your lab assignment tonight or Thursday, November 5th by midnight, just like normal, just like we were in class. The deadlines will remain the same throughout the semester, so just keep in mind of that. We are just now showing you it online. Lab 13 will be done by Professor Foss. So if you're hearing me right now and you're like, who is this random lady? My name is Danielle Dirks or Miss Dirks, Professor Dirks, whichever you want to call me there. And I'll be doing or talking to you about fungi today and leading you through your lab. So to get started, make sure, pause this video and go complete question number 1. It's about safety, laboratory safety, just like normal. So make sure you are explaining why, answering all parts of your question. Pause this video, complete that, come on back once you're done. All right, so this topic has been hit multiple times, but it is obviously a very important topic moving forward and for your final exam and probably the rest of your biology classes from here on out in college. So prokaryotes versus eukaryotes. I did a table here to kind of compare and contrast. So prokaryotes, examples are bacteria and archaea, whereas eukaryotes are fungi, protozoa, plant cells, and animal cells. Prokaryotes are much simpler in structure, whereas eukaryotes are usually larger and much more complex. They do have an internal membrane system, which allows for compartmentalization of different cellular and chemical processes, whereas the prokaryotes don't have that. They're much more simple, do not have that internal membrane system. Prokaryotes have a rigid or a semi-rigid cell wall surrounding a plasma membrane, whereas eukaryotes, some have cell walls, but not all eukaryotes have a cell wall. They all have a plasma membrane surrounding their cytosol, but not all eukaryotes have a cell wall. I know that question has shown up, I think, on both of your exams, and it was a tricky one. Make sure you know that not all eukaryotes have a cell wall. Prokaryotes have circular DNA, which is not within a nucleus, whereas eukaryotes have linear DNA, and it is compartmentalized in the nucleus. That is the organelle that has your genetic material. And again, that is part of that internal membrane system. Last but not least, they are similar in that they both have ribosomes, which help with protein synthesis. Moving on, fungi. So again, eukaryotic cells. There are two different types, yeasts versus molds. And then a fungal infection or infection caused by a yeast or a mold is a mycosis. Yeasts. There are typical yeast cells and dimorphic yeast cells. Typical yeast cells are unicellular, oval-shaped, whereas dimorphic yeast cells can grow as those unicellular oval cells, or they can also grow as filaments, which we'll see is similar to what molds do. Molds and filamentous, aka filamentous fungi. So they grow as long thread-like cells and spread via spores or condia. Examples of some molds or infections by these molds or filamentous fungi are they growing on old food. So you might go into your refrigerator a few months later, forgot you had, I don't know, some broccoli in there, maybe something else. Mold is what's growing on your food. Ringworm, jock itch, and athlete's foot, all caused by filamentous fungi. There are two types of filamentous fungi, septate molds, which septate molds have distinct tubular cells that make up their filaments. You can see that in picture A. And they also make spore-like structures called conida, which grow on the ends of the filaments. We'll see that in some of our slide photos later. Aseptate, which is seen in B, They are not divided into filaments, and they make spores, which are stored in the sporangium, or their chamber that stores those spores. So now I'm going to do a little bit of a microscope how-to, because if you were in class, you would be using your microscope, but this has also been a topic that has come up on quizzes and exams, so make sure you know this. It is important going forward in your biology classes that you know how to properly use a microscope. So first of all, super important that you know how to carry it and you're safe, because these are expensive tools and you do not want to have to replace them. So you carry your microscope to your desk station with one hand supporting the base or the underneath side of the microscope and the other hand holding the arm of the microscope. When you get there, everything should be set up for you if the last person did it perfectly. And if that is the case, first you will start by focusing your sample on the lowest magnification. So that will be at the 40x total. So the lens of the microscope itself is 10x, or 10 magnification. So this first, the lowest magnification you can go is by 4. So 4 times 10 is 40 total magnification. Once you've found and focused your sample on that lowest magnification, you can turn the knob to the medium magnification, where you'll focus it again there. That is 100x total magnification. The lens itself, again, is 10. By this medium magnification is another 10. So 10 by 10 is 100 total magnification. Next, once you've done that, you can focus your sample on the highest magnification, which is 400 total magnification. So again, that is by the 10 times the 40, x is 400 total magnification. Once you have focused in on your sample and you've gotten the data or the picture that you needed to get, that is when you are done, and we will start to reset it back up for the person that gets the microscope next. So always lower your stage completely to the bottom position, and then turn the magnification lens back to the 4x, or that lowest magnification position. Then you can wipe the lenses with that lens paper, and that is specific paper that does not scratch the lens. So when you find your microscope, you should find it completely with the stage completely at the lowest position and in that lowest magnification state. All right, next you will pause this video, observe the posted pictures for these 3 different samples, and then those will correspond to question 2 through 4. So complete question 2 through 4, then come back to this. All right, next step, watch the video of the wet mount procedure. This is one of the procedures that we will be missing out on, but I believe we've done one earlier in the semester. But watch that for a refresher on how to create a wet mount, and then view the photo of the wet mount that I posted. That would be, you would have done, looked at your wet mount underneath the microscope using the steps that we talked about earlier. And then that corresponds to question number 5. So complete question number 5, pause the video, come on back. All right, now go watch the cellophane tape method and view the photo of the cellophane tape slide, complete question number 6, and then come on back. All right, so fungal infections or mycoses, as we discussed earlier. There are 4 different types. Superficial mycoses, or tinea, is another name for that. They are caused by dermatophytes, which are filamentous fungi. And when those come in contact between moist skin and the spore or conidium of the fungi, that's what causes superficial fungi. And that occurs on the mucous membranes, skin, hair, nails. That's typically what you think of as athlete's foot, jock itch, ringworm. The next type of fungal infection is a subcutaneous mycosis. So subcutaneous means below the skin. So that is caused deeper in the skin, and occasionally underlying organs. That is caused by yeast or molds living in the soil, and they usually get into or under the skin via a wound or a puncture. The next one is systemic mycoses. So this is when your organs and or underlying deep tissues become infected by a fungus. It's important to note that this is very rare, but it's caused by inhalation of spores or filaments that are naturally occurring in the environment. So soil or spores that are within the soil, if those happened to be picked up via inhalation, maybe there's heavy winds in an area where they naturally live, that's where that can happen. Last but not least is opportunistic mycoses. These are caused by naturally occurring yeast or molds of the microbiome. So opportunistic means that normally they would not cause infection. However, something is off, something is altered, that they now have the opportunity to create an infection. So this happens in someone who may be immunocompromised or someone who has an altered microbiome. This can happen when someone is taking antibiotics, maybe because it destroyed a type of bacteria that is normally within our microbiome that competes with the yeast or fungus. And now now this fungus has the opportunity, since that is no longer around, to flourish and to create an infection. Or this can cause by, again, immunocompromised patients. This can happen in immunocompromised patients. That can happen with transplant recipients because they are given immunosuppressant drugs so that they do not reject their transplants. But this can wreak havoc when maybe there is a fungus around that now has the ability to cause infection. So that is something that needs to be monitored closely for transplant recipients. Additionally, those with HIV or AIDS, and HIV causes a disruption in the immune system, which then allows these infections to cause problems for those patients. This again can happen through inhalation of cells or spores, and it's important to note that this can happen as a superficial, subcutaneous, or a systemic mycoses. All three of those can happen. Some examples of these are Candida albicans, which is typically what causes candidus or thrush. This can happen when someone's immune system suppressed, again, an increase in estrogen, or someone is on antibiotics. And then another example is a dimorphic yeast, actually, and it causes cryptococcosis, and that is typically seen in transplant recipients and or people with HIV and AIDS or AIDS. Antimycotics / antifungal agents. So these are what destroy mycoses causing fungi. So they're like antibiotics, but for fungus. There's both topical, which is used to treat superficial mycoses, obviously, and oral or intravenous, which are used to treat more systemic infections. Examples of those are listed below, but a little fun fact: if you see something that ends in AZ, OLE, or azole, that is typically an antifungal agent. Now that you know this information, go complete your case study or question number 7. Let me or Professor Foss know if you need any help. We are here to help. Just because we're online doesn't mean we are not a resource to you. Please email us with any questions. make sure you don't do it too late in the day, because we do like to sign off and have some time with our families as well. So get on your lab report, turn it in on time. Don't forget. Talk to you guys later.